Welcome, dear students. Now I will be dealing with cytoplasmic male sterility. You see, this is a type of male sterility which is controlled by a cytoplasmic gene known as S gene. So if you see that it is result of mutation in the mitochondrial genome, spontaneous mutation you can say, which leads to mitochondrial dysfunction. This results unfavorable nuclear mitochondrial interaction resulting male sterility. Hence, male sterility is controlled by cytoplasmic gene S. Here you see the CMS. The capital S is in the cytoplasm. As you know that mitochondria is in the cytoplasm. So if you see the origin, of course, it is also originated through spontaneous mutation in many crops, in many plants it has been observed. It is occurring due to a spontaneous interspecific hybridization, It can also be created through induced mutation, and it has been observed that ethidium bromide has been found to be most effective for mutation in cytoplasmic genes, which have been done in many crops like pearl millet, barley, etc. Or it can be transferred from known sources, as I have described in my back cross breeding program and somewhat in my CGMS system lectures. So if you see the inheritance, you see this is the CMS, where you are seeing that the nuclear gene is normal, but in cytoplasm capital S is available. If you are crossing with another variety which is normal, is not having fertile cytoplasm, then the F1s you are getting. In F1s, as you are knowing that cytoplasm is coming from the female, that is why all the hybrid seeds obtaining from this CGMS system will be male sterile. So how can you use it? Uses. This can be used for hybrid seed production in ornamental as well as in crops where vegetative parts are of economic importance, because that if an hybrid progeny will not give the seeds, will not produce the grains. So in grain crops you cannot utilize the CMS system. For that you have to go to CGMS system. As you know, CGMS and CMS, there is no difference. Once you are knowing the restored genes in the crops, the same CMS system becomes CGMS system once you are utilizing the fertility restored pollinators. So this is all little about the cytoplasmic male sterility. As I said, that many more things of CMS have been described in my earlier videos of CGMS system as well as back cross breeding. I have described many things. As well as if you see, as I have already discussed, while the types of male sterility, of course, we have discussed genetic male sterility. In that, the genetic male sterility, I have described many types. The general type of genetic male sterility, the EGMS, in EGMS, PGMS, TGMS, then TRGMS. These are the GMS type male sterility. Then I have already described this cytoplasmic male sterility. Of course, this system has been described more in my third type, CGMS, cytoplasmic genetic male sterility system. Then another, apart from these male sterility, you can use chemical hybridizing agents also for creating the male sterility. Once you are not able to get all these, so you can go for chemical hybridizing agents. You can spray the chemical hybridization, the agents, the chemicals. Your whole one parent will become male sterile, and another parent you are not spraying the CHAs, that will be normal. So whatever pollen grains are coming from the normal plant, normal variety, that will be pollinating and fertilizing your male sterile created by CHAs. So whatever seed you are getting the hybrid seed. If I am getting time later, I will be dealing the CHAs also. But here I tell you, in India we are not using CHAs for creating male sterility and utilizing it for hybrid seed production program, because majority of the CHAs are hazardous to the health, to the environment. They have carryover effects on their next progenies, which we are going to use as grain. So to avoid all these things, we are using these male sterility system in our hybrid seed production program. So of course, if I am getting time, I may be dealing the CHAs also. So these are the types of male sterility I have already covered. Thanking you very much.